As a reminder, these videos are a condensed version of the videos the College Board is putting out for AP Bio Review. So I highly recommend checking out those videos for more in-depth explanation. We're going to start with the properties of water. So we learned in class that water is H2O, 2 hydrogens, 1 oxygen, and it is a polar molecule, meaning that it's partially negative on one side and partially positive on another side. And you'll remember that oxygen is the partially negative area, hydrogen is the partially positive area. This causes neighboring water molecules to be attracted to each other. The negative, partial negative, near the oxygen of one water molecule is attracted to the partial positive of the hydrogen of another water molecule. We call this attraction a hydrogen bond. Hydrogen bonds are relatively strong. They hold water together, but I want to remind you they're not as strong as the covalent bond that holds the water molecule itself together. The reason that oxygen is partially negative is because oxygen is what we call electronegative. That means it is like an electron hog. It holds the electrons that are part of the covalent bond really close to itself. So it ends up with more of a negative charge because the electrons are negative, and the hydrogens end up with more of a positive charge because they're left with just their proton. This partial negative and partial positive and hydrogen bonding leads to key properties of water that we learned about. For example, cohesion. Cohesion is when water molecules are attracted to each other like we see in this picture. Cohesion leads to surface tension. Surface tension is what allowed us to balance the paperclip on top of the water in the Petri dish in class. It also is what is responsible for the amount of water that we were able to put on the penny when we dropped water on it in class, and we got that big bubble. That's the cohesion of the water molecules pulling on each other. Another property of water is adhesion. This is water's ability to stick to other things that have partial charges. So for example, the sides of the xylem in a plant that is the passageway through which water gets all the way up through the plant. Additionally, evaporative cooling and heat capacity are two other properties of water. Evaporative cooling means that the water molecules moving the fastest are the first to evaporate, leaving the remaining water molecules in the sample to be moving slower, which ends up decreasing the temperature of that sample, because heat is all about how fast particles are moving. Heat capacity is the reference to water's ability to buffer its temperature. So, for example, if we were turning this sample, which is possibly liquid because these water molecules are pretty close together, if we were trying to turn it into a gas, or if we're adding heat to it, rather, when we're adding heat to it, some of that heat goes to breaking the bonds between the molecules. And so instead of the water molecules themselves moving faster and being hotter, we've used some of that energy to break the bond, and so the sample's temperature doesn't go as much as the heat that we've put into it. Also, a property to remember is that ice floats. This is because when water molecules arrange themselves in a crystalline structure, they end up being further apart than they are when they're in a liquid form and they're kind of flowing past each other. This is pretty unique to water. Also, water is a universal solvent because it can dissolve anything that has a charge. So for example, salt, NaCl, has a positive charge on the Na and a negative charge on the Cl. That's an ionic compound. It splits apart. And the water molecule can surround the Na and surround the Cl and dissolve the salt into the water. Other chemistry of life concepts. Don't forget about the elements of life. So they are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. These are the most abundant elements in living things. And as a reminder, this is what elements are in each of the 4 major macromolecules that we learned about. So carbohydrates have carbon, hydrogen, oxygen. Proteins have carbon, hydrogen, oxygen again. You'll notice they all have those 3, plus nitrogen and sometimes sulfur. Notice sulfur only happens in protein of these 4 major macromolecules. Lipids have carbon, hydrogen, oxygen. Nucleic acids, again, carbon, hydrogen, oxygen, nitrogen, and phosphorus. And phosphorus is unique to nucleic acids in terms of these macromolecules. So remember that. Nucleic acids, phosphorus, proteins, sometimes sulfur. More about those major macromolecules, also known as biological molecules. So most of them are made up of monomers, and when we put multiple monomers together, that's called a polymer. For example, carbohydrates, the monomer is a monosaccharide. For example, glucose is a monosaccharide, it's one sugar. Other types of carbohydrates are starch, cellulose, and lactose, and others. So carbohydrates are when we have a chain of multiple monosaccharides, multiple simple sugars assembled in a line, and the way they're assembled leads to what type of carbohydrate it is. Protein. The monomer of protein is an amino acid. Here is one amino acid. Reminder, there's a central carbon, a carboxyl group, an amine group, a hydrogen off of the central carbon, and an R group. An R group, R is not an element, R stands for like X, it's like a variable. Each amino acid has a different R group, a different set of elements in that space. And proteins are assembled, multiple amino acids are assembled in different orders to make a protein. And we'll talk more about that in the future. Lipids are a little bit different. They're not always a monomer assembled to make a polymer. Sometimes it's maybe 3 fatty acids and a triglyceride, but what is always consistent is it's always carbons and hydrogens. Sometimes there's oxygen, but it's typically a long chain, so you'll see something like this. There's saturated and there's unsaturated. Saturated usually means straight. It means all of the carbons have a hydrogen. That's the saturated aspect. Unsaturated means not all of the carbons have all of the possible hydrogens, so they have a double bond, and that results in a kink in the, like a bend in the fat. As a reminder, lipids are all nonpolar. Nucleic acids, the monomer is a nucleotide. It's made up of a phosphate group, a sugar, and a nitrogenous base. This makes up DNA and RNA. As a reminder, proteins are things like enzymes and protein channels. Lipids could be like phospholipid bilayer or steroids or hormones or cholesterol. Those are all examples of lipids. And while we're talking about macromolecules and how they're assembled from monomers into polymers, let's talk about how we put those monomers together and how we separate them again. So this is hydrolysis and dehydration synthesis. Hydrolysis, the word breaks down to hydro, water, lysis, break. So we're breaking a bond and we're using water in the process of breaking that bond. So for example, I have 2 sugar molecules here. Sometimes you'll see them just abbreviated as a pentagon like this. They have an oxygen, 2 covalent bonds going to that oxygen, holding them together. In hydrolysis, the atoms from water are used to break that bond. Plus you would need some energy. But you'll notice that the 2 hydrogens end up as part of the sugars, and the oxygen from the water also ends up as part of the sugar. The opposite is dehydration synthesis. Let's break that down. Dehydration, remove water, synthesis, to put together. So in this example, I have amino acids. This is true of all of the major macromolecules, the hydrolysis and dehydration synthesis. I'm just using different examples. Here I have 2 amino acids and I'm putting them together, hence dehydration synthesis. You'll see that the O, H, and H are removed, water is removed, and we end up with a covalent bond between this carbon and this nitrogen. So dehydration synthesis is putting molecules together.